Okay, today I wanted to go over with you in detail the scoring for the Unit 6 Practice FRQ so you can better understand what is expected of your answers for the types of questions you'll receive not only on the AP exam but in the next exam, in the Unit 6 test. So let's go ahead and look at question number 1. And on the surface, it appears to be a pretty simple question. And truthfully, it is. However, the biggest problem people had was details. All right. This one happens to be worth 8 points. So this is what we would call a long FRQ, which would take around 20 minutes. And so this means they expect you to go ahead and give more details. Now, it wants you to contrast the following activities in prokaryotes and eukaryotes. Now, in setting up this answer, it gives you 4 basic categories. Now, what that means is it wants you to hit all 4 of those categories. And so you want to be very specific. One of the points that I made on a lot of people's FRQs when I went ahead and scored them. And if you haven't looked at the detailed notes I gave you, I gave everybody detailed notes to make their answers better. So what you want to do is you want to go ahead and address these one at a time in specific detail. So let's look at the breakdown of the points. There's 4 categories. Now, the rubric basically breaks it into 5 because this could possibly be giving as many as 4 points. Now, each point means you had to get the bullet point in both places. So, for example, if you're talking about DNA replication, telling me prokaryotes have a single origin, well, eukaryotes have a multiple origin replication style, that gives you 1 point. Now you need to hit at least 1 in each category, and then it says you can have each box is worth a maximum of 2. So if you look at this, there's a potential of 10 points and you only need 8. You could get 2 from this box, 2 from this box, 2 from this box, 2 from this box, and 2 from this box. So when we look at this, most of these should be self-explanatory. In the DNA replication, the second one is the presence of telomeres. And the third one, the location that it occurs, the nucleus versus the cytoplasm. Then let's talk about transcription. Transcription is the making of messenger RNA from DNA. And so in prokaryotes, there's no processing. In eukaryotes, there is processing. Location for prokaryotes, cytoplasm. Eukaryotes, the nucleus, mitochondria, and chloroplasts. Now, the term monocistronic and polycistronic. I know those are advanced words, but what they mean is that in a prokaryote, 1 sequence of DNA, 1 gene equals 1 protein. In eukaryotes, 1 sequence of DNA, because it is processed, can give rise to several different proteins in the same gene. Initiation factors, sigma versus regular initiation factors. That's a really minute detail. Most people don't go over that, not a big deal. Also the variety of polymerases. Now in translation, this is going DNA to protein. I was very happy to see most people's, or at least quite a few of you, got the transcription and translation are one process in the prokaryote. They go feed one from the other, and that they are separated in the eukaryote. The next part, 30-40 and 40-60, those are the subunits of the ribosomes. The ribosome structures are different in prokaryotes and eukaryotes, and we did cover that. Location, once again, you have to be specific. You can't just say the location is cytoplasm versus something else and expect it to apply to everything. You have to specify for each process. Gene regulation, prokaryotes do not use operons. Gene regulation in prokaryotes, prokaryotes use operons where eukaryotes do not. This is like the lac operon that we were talking about. So prokaryotes can use positive and negative control where eukaryotes cannot. Location, again. Oh, I'm sorry. Enhancers are rare where enhancers are common. This would have to do with the ligands available to encourage the production of proteins. For example, those that would be produced by the RAS gene. Then we talk about actually gene regulation. The whole gene works for the prokaryote. It doesn't shut down portions where methylation, acetylation, and bar bodies all are part of the regulation of the eukaryotic genome. Cell division, they just have binary fission. It's rapid, no spindles. In eukaryotes, it's mitosis or meiosis, depending upon the cells produced. It's slower, and you need spindle fibers. So, hopefully you understand your answer if you look over my notes and then see what they are looking for. This is the College Board rubric, so that if you were being graded for your AP exam, this is what the graders would be looking at. Next one, so there's a total of 8 points. Sorry about the big blank spaces, but I had to take out some of the extra stuff from the AP College Board. This one talks about flies. These are perfect flies. They have no genetic engineering. No genetic variation. They've been engineered. But how could variation appear? Now, this one you guys pretty much hit out of the park. You could use any of these reasons. Now, please don't just say sexual reproduction, because sexual reproduction, what is it about sexual reproduction? Is it crossing over in the production of gametes? Is it the random assortment, random fertilization? You have to be specific. You can't just say sexual reproduction. All right, so that was 1 point. Next one, how can a single base pair mutant in DNA alter the structure? And here's the part that most of you guys miss, the function of the protein. You can't say the front function changes. You have to say specifically how and why. Now, this was worth 4 points. Now, you got 3 of the points. Most people got 3 of their points right here. Now, you didn't have to, but here's where you got 3 points. Most people got their 3 points there. You could get an additional 3 points from the protein alteration. Now, for a total of a maximum of 4 points. Now, it was easier to define the mutations. Now, be very specific as to what you are talking about. People just used codons without explaining it, base pairs, nucleotides. Make sure you specify your vocabulary words. After that, the biggest problem was the protein. One way you could get the protein point, because there's 4 points and a maximum of 3 can come from one of the areas. You had to describe that the protein structure, the different types of protein structure, and that it would alter that. The other ones, it could change the active site in the end protein. And so that could cause a protein not to function. An example of this would be the video we saw on diabetes. You could also talk about the problems with the bonding and the folding, and that the amino acids, because that they are different in the protein, would cause different bonds and alter the shape. 4 points max. Next one. Here we go. This one was as easy to set up of an answer as there could be. Now, one of you guys, hi Ren, actually went and bullet pointed each one and said, RNA polymerase does this, lysosomes do this, and that was perfect. This is exactly how you should set up this question. Now, so what they're looking for, what were the specific jobs for each one? RNA polymerase. it's going to make messenger RNA. And I would specify messenger RNA there from DNA. Although it could do some other, that's if you can specify, do it. Spliceosomes, it removes the introns and connects the exons in RNA. And if you can remember, specify messenger RNA. Now, this, it isn't always the case, but sometimes you will lose points if you don't specify what type of RNA. Codons, they code for specific amino acids. And then you could have said something about the triplet code, that's fine. Ribosomes, the RNA, this is the site of protein synthesis, and transfer RNA, this is the specific type of RNA that transports the amino acids to the ribosome during translation. Total of 5 points. Now, this was a really good question. I like this one. Proteins can be produced either in the cytosol or attached to the ER. When they are attached to the ER, they tend to function to produce different substances than if they are in the cytoplasm. And so you had to say which one you're talking about and what is the function. You didn't have to be specific about the protein, but you had to give, how should I put it, specific generalities. For example, cytoskeleton pieces. That would be general in the fact that it doesn't tell you the name of the protein, but specific enough for its function. So you had to go ahead and you had to pick, you basically had to have 2 pieces of information. You had to say where it went, and you had to say what the function is when it goes there for each of the 2 situations. Now, there was a lot to pick from. So, first one, ribosome. If you chose the destination excreted from the cell, here's your 2 choices. If it was in the integrated cell, in the cell membrane, here are your choices. And that's how you read this question. There's a lot to go through. You had a lot to pick from. If it was inside the Golgi apparatus, you had to go ahead and pick one of these functions. Then you had your second. If it was in the cytosol, here you go. If it's in a vacuole, here you go. And if it's in the nucleus, here you go. So you got 2 points for each pair for a total of 4. Finally, describe 3 different types of mutations and their effect on protein synthesis. All right, so here's the thing. You should name the mutation. Please name your mutations. 2, explain what it is. 3, the effect on DNA. And then 4, the effect that has on the protein. You have to go in, basically, it says describe 3. Now, part of the reason they say describe 3 is because they're going to assume people they'll miss a detail. They're giving you basically 3 chances to answer for 2 that they need to see. So, they give you a whole list of mutations, and you have to say exactly what it is and what the effect is. So, let's look at their list. You had to pair these up. So, here are your possible pairs. You can have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 different types of mutations have been covered. You had, in order to get all 4 points, they asked you to describe 3, but you only needed to get 2 correct to get full points. They do that a lot, so make sure you hit them all. Sometimes they do that a lot, and sometimes they don't tell you they want more detail. Give as many as you can in the allotted time, especially if it's a long versus a short. Now, This gives you an idea of what the effect is. So you had to make 2 pairs. Please note that frame shift mutations were all one type. Insertion and deletions are the same type. Some people broke those 2 up into the second and third, but because of the way the scoring was designed, you still got full credit. But please know that frame shift has 2 types of frame shift. You could pick from any of these, and this includes the ones in gamete production, not just the ones we covered most recently. All right, so that gave you your 4 points, and that is the end. You guys did really well. Basically, I had, most people were scoring 4s and 5s. So you guys did a great job. I've given you a copy of this scoring rubric. You can look over it, and if you have any questions, that's a great thing to bring up in our study session. So study for your test. It's going to be a tough one. Make sure you know your vocab and get all your details. I'll see you guys tomorrow.